All right, everyone, welcome back today. We have an extremely special guest, probably one of the most prolific researchers in the aging and longevity field, Dr. David Sinclair. Give it up.
Hey
He's a
everyone.
very busy man. He couldn't make it in person, but I think he's still going to deliver something extremely interesting today. So let's pay extra attention.
So I have about 20 minutes, yeah?
Yep. And if you have time after, you can ask some questions. And then Gladyshev is joining us after in person.
Great. I was just talking with him. He's great to have too. Good. Well, unfortunately I couldn't be there today. I'm here at home with family. But yeah, I wanted to drop by. It sounds like what you're doing today is an unusual thing. And I thought I'd join in on that and present some unusual things. And what I'm going to present to you today is something. A lot of these slides I've never presented before. I pulled them out of the archives about 2014. I had a phase where I was just writing down notes constantly, drawing slides, drawing equations, and I've never shown them in public before. They led to Lifespan, the book. They've led to pretty much all the work that we've been doing in my lab since. And I still use these thoughts to drive my lab's hypotheses. And it's a very different way of thinking than most labs when it comes to aging. And I'm happy to share with some of them with you today. And what I was actually trying to do was to come to grips with a lot of experiments that weren't making any sense at the time. That's often the way science goes, right? You have a paradigm shift that's necessary because the current paradigm just doesn't make any sense. And some of the things that weren't making any sense were that you could increase DNA damage in a mouse and it would live a normal lifespan. And But in my lab, we were creating special types of DNA damage and we were accelerating aging. So how do you reconcile that? What is it about these sirtuins that we've been working on since I was at MIT? And what is it? Is it really just that we break down, or is it something more than that? And through a wormhole, a deep dive, I would say, I got really deep into information theory. I got deep into the work of Claude Shannon and his colleagues, and even earlier. And things clicked. They started to really make sense to me that aging isn't so much a linear process, it's a circular process. And that aging is unlikely just to be wear and tear, but it's actually a software problem. And I began looking for ways to test that. And we still continue to test that. There are times in my lab where still have confusing results, and we're challenging this hypothesis all the time. But compared to 10 years ago, we are much more, I should say, I'm much more enthusiastic about it because it really is, it's explaining a lot of things that have been going on, not just in my lab now, but globally. And you'll hear more about that from Vadim about epigenetic clocks. And we've been working in my lab on epigenetics since day one, and even before I had a lab. And so let me share some of that with you. This is going to be the first talk, I don't know, maybe ever, that I'm not going to show any data. I'm happy to tell you about what we're up to and where we're going and what the latest is. And I have some slides, but I decided this is a different group of people, different meeting. So let's talk about something that's a little bit more high level than messing around with cells and mice. You'll probably get a better insight into my brain than ever before. So this is an old yeast cell. I took a picture of it when I was in my 20s. I was working with Lenny Guarente, who I owe a lot to, my professor at MIT. And this is an old yeast cell. And what Lenny and I co-published in Cell in 1997 was that two major things happen for yeast aging. One was it seems to be somewhat of a program in that there's a stochastic element where you get genomic instability, the DNA breaks, you get a circle formed of ribosomal DNA. But after that event happens, now it's a very predictable program. We could mathematically model and predict the lifespan of yeast cells and their offspring. And all that math is actually buried in the back of that paper. And what we proposed was that the genomic instability in this organism led to the distraction of gene regulatory factors, and it was messing up the epigenome. And that gave rise to the phenotypes of aging, not just the death, but the phenotypes or the characteristics of aging in yeast are similar to us. They get old, they get slow, they get large, as you can see with this yellow acidic cell stained with bromophenol blue. They become sterile. And we could change the lifespan of a yeast cell by creating these circles of DNA or breaking the DNA in just a few defined places that would accelerate aging, including the sterility and the size and the slowness. And we could do the opposite. We could actually slow down the formation of these. And other colleagues in the lab made mutations that prevented circles from forming as frequently. And as the model predicted, the yeast cells lived longer and maintained their fertility. And so that led to a real shift, because at the time in the 1990s, when I arrived at MIT, the idea was that aging was caused by damage. So we expected in genetic screens to find antioxidant enzymes, but instead we found genes that controlled other genes, silencing proteins, which we now call sirtuins. There's a lot known about sirtuins, and there's not a lot of people, but there's a few people who still claim that it's debatable. It's not really debatable. We know how these enzymes work. We know how to activate them with small molecules, as you can see in this review by Michael Bunkowski and I. And so, I've spent a lot of my career trying to help people be healthier. So I've been co-developing molecules that can activate these enzymes. So you don't just have to fast or exercise to get the benefits. So these enzymes, what they do in yeast and in humans, in large part, is they stabilize the epigenome and they repair broken chromosomes. And they actually physically move, we think, between silencing and controlling genes and repairing DNA. And that's important for the hypothesis too. But what's important about sirtuins, named after the original SIR2 gene, is that SIR stands for information. And so that's obviously been driving my quest to understand what is it about information that's relevant to aging. So this is a series of slides that I drew in the early teens of this century. And so, these are ideas. These are not published. You can see what kind of things that I was thinking about at the time. And I was thinking, maybe we just need to revise the old paradigm. And instead of thinking about stages of damage leading to aging, maybe aging really is a circle, and that it's malleable, and we can not just accelerate it, which we were doing at the time. It's all published now. But we could even reverse that process. We could reset the epigenome if that's truly involved. And you can see here, I was writing things like resettable epigenome, and we were looking to use the Yamanaka factors potentially to do that. And of course, Juan Carlos Belmonte deserves huge credit for being the first to do this in vivo, to show in vivo you could actually very carefully treat a mouse with Yamanaka factors and not kill it, and actually make it healthier and longer lived. And we have stood on his shoulders as well. I'm fascinated with cell identity. Of course, the epigenome gives cells their identity. It's what allows development to happen. It's how you get an embryo from a single cell. And the idea at the time was maybe we get what I came up with at the time was called epigenetic drift, that due to insults to perhaps the chromosome, DNA breaks, circles of DNA. These distract the epigenetic regulators, and what were once very powerful boundaries and accurate gene expression levels become smooth, and actually you don't get the beautiful pattern of gene expression. And I did some mathematical modeling of that helped my thinking. And then, inspired by Claude Shannon's work, I started going nuts about analog versus digital information and noise, and the observer, which you'll see later, and started to model biological noise uh, in the same way that Shannon modeled informational and digital noise. And I started to realize there were two types of information in the body. There was digital information, which is DNA, and a lot of the epigenome, which is just as important for life and longevity, is analog. Analog information is stored in the body as structures of how the DNA is actually wrapped. And it's in 4 dimensions. It's not in 1 or 2. And I started to think about correction data. Was there correction data really stored in every cell? And we started to look for it. And we were wondering, how do we go back, like Claude Shannon suggested, to not just slow down the noise, but actually reset the information in the cell. Here I'm saying maybe it's stored in the form of RNA and DNA. And the output of all of this system is life and longevity. And then I started to think about what makes us different and similar as species. And this was an idea where the epigenetic landscape, which gives beautiful gene expression, this is, of course, modeled after Waddington's landscape, is that current interventions are trying to slow down this bouncing of this virtual cell that's not trying, but can jump across valleys. But it doesn't when we're young, because these boundaries, epigenetic boundaries, which Manolis has beautifully defined, they erode over time, we think partly due to cell stress and broken chromosomes. Now we've got more entropy, less boundaries in the epigenome, and now you can see that cells can jump from one cell type to another. They don't really jump, but we do see them drift. And actually, experiments that we've now published show that older cells are easier to reprogram into different cell types because the boundaries of their cell type are lower. And perhaps I was thinking at the time, perhaps a species that lives a long time, like a bowhead whale or a Greenland shark, would have less vibration, fewer DNA breaks, but also greater stability of the boundaries that keep cells and maintain their identity, so that even an chronologically old Greenland shark, compared to us at the same age, at let's say 90 years old, would have greater ability. Well, perhaps here is what we would compare to this, has greater boundaries. And so I was looking for ways in my lab to slow down this process by increasing sirtuin activity, increase DNA repair, protect the genome. We've now done that. Some of it's published. We have enzymes now and proteins that stabilize the worm genome and make worms live longer. We're doing this in mammals as well. But what about, can we truly slow down the erosion of these landscapes? And can we ever get this cell to go back to where it was? Is that information even possible? Now, making a stem cell using Yamanaka factors was not going to be the solution, because importantly, the difference between what Yamanaka did and what I'm trying to do here is that Yamanaka erased cell identity, basically took these hills and flattened them so that the cells had no identity. I'm trying here to do the opposite, regain the identity. So I started to call this process X-differentiation. Richard Cutler has called it dis-differentiation. And then the redifferentiation would be the reset. And this is a Waddington's landscape of the epigenome during development. And in this review and perspective with Wencheng Liu and Xiao Qian from my lab at the time, we've extended the Waddington landscape out into the future. So now it includes the adult and the old individual. And you could see that the cells that were once in a beautiful valley are now more likely to move around the landscape and lose their identity. And losing your identity is a real problem, right? You're probably more susceptible to diabetes, Alzheimer's, even cancer. And we've written about that as well. For cancer, it's called the gerontogenesis hypothesis. So this is more of a fully formed depiction of the theory that we've published. And of course, if anyone has read Claude Shannon's mathematical theory of aging papers in 1948, parts A and B, they will recognize this. I have stolen it and just replaced some of the boxes with biology rather than math and numbers. And it's similar, though, that our bodies are like information that gets transferred across time and space. There's noise that's introduced by damage to cells, and eventually the information is lost. And most of it is epigenetic and analog information, because analog information is much harder to maintain over a lifetime than digital. Of course, you'll appreciate that that's the reason we live in a digital world. The major reason is that it's less subject to noise. And even if you do lose digital information, you can go back with the TCP/IP protocol and find the missing bits with what Claude Shannon was calling the observer. Now we call it the backup copy. And we've been looking in my lab for, first of all, how do you contact the observer and what is it made of? Where is epigenetic youthful information stored? And we have evidence that it exists, which is really exciting. So we use this paradigm to cure blindness in mice. We're now improving vision in blind monkeys at Life Biosciences here in Boston. And hopefully we'll, if all goes well, treat a human for blindness next year. Or if perhaps even multiple diseases, not just regular blindness, but perhaps glaucoma, macular degeneration, perhaps one day an entire human. But we're starting with the eye, if you're wondering, because for gene therapy, it's the best place to start. It's an enclosed, immune-privileged location in the body. One of the papers we published that was really inspirational for this theory, or driver, was what we used to call this theory, but it was a terrible name. It was called the Relocalization of Chromatin Modifiers hypothesis. And it was a 2008 Cell paper by Philip Oberdorfer, postdoc at the time, and of course many collaborators. And what we found was that in vitro and in mice, we could create double-stranded breaks and see the movement of epigenetic regulators, including the sirtuins. Now we know SIRT1, SIRT6, HDAC1, other labs have shown this. They move to the break. Gene expression changes in response to this over a matter of minutes, but not all of the factors go back to where they came from. So that the genes that are here over time become deregulated. Even if the DNA is repaired 100%, there is an epigenetic scar because of that previous DNA damage, which is important to remember, right? Because we're so trained to think DNA is our destiny that even if DNA isn't mutated, it can have a lasting effect on an organ. And our cells experience a few DNA breaks every day, depending on the cell type, of course. Sometimes it's more than that. I fly a lot, so I have more than that. X-rays, CT scans will accelerate that. We forget that even if our body is 100% effective at repairing that, it doesn't mean that the cell is the same as it was. This is from that same review, and I want to use this to re-emphasize the point that we're not trying to make stem cells or go anywhere near a stem cell. It's often misunderstood. This theory is often misunderstood that what we're trying to do is push cells into a stem-like state, which allows them to then be healthier. We don't see evidence of that. In fact, what we think we're looking at is something in between, which is the cells regain their identity, their function. But if you push them too hard, like Yamanaka was doing in vitro, Then you go back to this early blue stage here of being a stem cell. But we're really looking at taking cells that have lost their identity and getting them back to being young, but not too young. And that was the trick. That took us years to do. And we almost gave up because we kept getting stem cells rather than differentiated cells. Interesting fact is that Nalat, a graduate student in the lab, is applying this theory to cancer cells. And it's often thought that we'll make cancer cells worse if we reprogram them because the mind goes straight to this stem cell state. But remember, we're trying to regain identity. And if you make a cell that's cancerous or oncogenic regain its identity, first of all, I think it's going to behave better. Second of all, what we're finding in practice, at least in cell lines, is that the cells in this process of redifferentiating, they kill themselves. It makes sense, right? You wake up from a zombie-like state and you see all the trouble that you're causing, and you probably want to get rid of yourself. So that's what we're seeing in cell lines. Just really briefly, one of the reasons that I'm enthusiastic about there being a repository of epigenetic information in every cell is this kind of data. This came out in 2020 and from Wan Cheng's paper. And this is a DNA methylation pattern across the entire genome of retinal ganglion cells, these nerves that go from the eye to the brain. And this is the pattern in the young RGCs. The old look like this. The negative control, untreated, is this. And the last 1, 2, 3, 4 mice, the pattern is remarkably similar to the young animals. So think about that across tens of thousands of sites on the genome, the cells are able to restore many of those methylation sites. The same is true for transcription. If we look at these same cells, the reason that those dots, which represent each one a locus, a separate RNA, it's not a perfect straight line, but it's pretty damn exciting that a gene that loses its expression by twofold during aging roughly gains its expression by twofold during reprogramming. And the point that I want to make is, how is that possible? Is it simply just transcription factors interacting with transcription factors, interacting with more transcription factors, which interact with DNA methyltransferases and demethylases? Maybe, but I'm betting that there's more than that. That there's physically at these genes, there's something that we're missing, a memory of the gene expression state and the chromatin state that cell should be in if it gets rejuvenated. And so why would it exist, you might ask? Well, I've written that this is likely to be a program that life has had since the very beginning to regrow, to rejuvenate. We have so-called immortal species like jellyfish and worms that can be cloned. We can even clone mammals. We could clone ourselves if it weren't illegal. And so how is that possible? How can you reset cells? How can you regrow tissues when they've been cut out like a liver out of a human, leaving only a small piece behind? I think there is a repository of youthful information that we're tapping into by, in this case, this experiment, we're overexpressing 3 of the Yamanaka genes, OCT4, SOX2, and KLF4. We leave out c-Myc, which is an oncogene. And we find it's very safe. This is the method we're going to use in humans. And then just lastly, for lack of time, I need to end, but this is the kind of thinking that we have, that there are tags at genes that have lost their gene expression. And they may be tags on the DNA, there might be tags on proteins, there might be RNA, they might be a new type of information. We're looking at all of this, and I've given some examples of what these lead candidates that we're going after might look like. When this pattern is laid down, I don't know. It could be during development. It could be during the teenage years when we're at our optimal. It could be a mix of all of that. But first we have to find them. And so right now, if you're wondering what we're doing, Chris Petty in the lab and others who are working with him are destroying these marks, hypothetical marks, and seeing if reprogramming is also destroyed. We're also enhancing the marks to see if we can enhance rejuvenation and reversal of aging. And this is my last slide, I believe, where aging is the loss of cell identity with age, and what we're doing in our work is taking a cell that's lost its identity and regaining it so that it can express those genes beautifully like it did when it was young, in a semi-permanent way. This is not just, do it for 2 hours and it goes away. We can rejuvenate a mouse's eye and cure blindness with Bruce Cassandra's lab, who's here at Harvard. And we've now published since the first paper that this effect lasts nearly to the end of the mouse's life. The mouse only dies because the rest of its organs are old, but its eyes are still really young, which is interesting to think about. We also know we can reset more than once. And it's a lot of brilliant people's work that I've had the fortune to present to you today. And I've mentioned people along the way, but I want to thank the people who have collaborated for the last, 25 years that have made this possible. Thank you. I'm happy to answer a few questions.
Give it up for David Sinclair. You have many fans in the audience, fans of you, Vadim, who's in the background, and Michael Levin and Carl Friston, and all these people that are sort of taking this more mathematical and sort of info-theoretic approach to biology. And if any of you have some really good questions, now is the time for David.
You mentioned Mike Levin. While you're picking people, I love Mike's work, and he and I have discussed, we're excited about the possibility of intersection between what we do and what he's discovering.
Wonderful. I want to have some questions. One in the back. Yes. Speak loud.
Okay. So my question is,
David, can you hear?
Yes.
Okay, go for it.
so is it possible to look at it as a cellular automator, like a neural cellular automator with stresses and rewards to the system? Is it possible to describe the system like this?
David, now's the time to talk about hormesis.
I think I understood the question. It was a little bit fuzzy. So we see a lot of cell autonomous effects with reprogramming as well. And the link to hormesis is that this program, if you want to call it a program, and actually I would call it a program because it seems to be, what do you call it? Reusing aspects of embryonic development, that this program is designed to respond to the milieu, the environment. And during development, of course, there's different environmental factors, often from neighboring cells. But as you become an adult, the environment that's super important is the liquid that you're around, which is influenced by how you live, what you eat, how much you've slept, how much you've exercised. And that seems to turn on these defense pathways in each cell independently. The sirtuins come on, for example. There are others, mTOR and AMPK are popular ones. They all talk to each other, so there's no one that's more important than the other, despite what the field likes to argue about. And so, yeah, it's a long answer to a question, and I worry that I didn't fully understand the question. But if you want to guide me and help me focus on one aspect, feel free to tell me.
He's actually working on a project inspired by you and Michael Levin for this event. So definitely let's.
Yeah, so I was looking at So, it's like, yeah. So I'm looking at like fractal expressions of like the gene, like trying to use like fractal calculus a bit, or like trying to like understand like if it's possible that the way this is acting is like it's trying to reach a thermodynamic state, like an equilibrium kind of, because like it has prior information. Like I'm basing on like this assumption that it has like a genetic like image of what it wants to be, and like it's trying to reach this like equilibrium, and I'm trying to explain it through like fractal math, and I don't know if it's like the right way to go about it.
Yeah. Well, I'm going to say something that might not be relevant to your question, but I'll say it anyway because that's what academics do when they're not sure about the question. I love these new life forms that sounds like you're working on and Mike has published on, and it's an important way to look at life. Too often we look at organisms as organisms, but cells are organisms, right? It's a very large colony. And because we've got consciousness, we think of this as a single thing, but of course it's not. It's very complex. But the cells are things, right? Cells want to survive. They want to reproduce. They like finding partners. They like interacting. They're very social. And I think what you're tapping into, what Mike has tapped into, is that fundamental aspect of life that's been with us since the first proto-organisms. And that's why in my book, I pretty much started it with the beginning of life, because I truly believe that understanding the beginnings of life help us understand life at this stage. And being able to recreate those interactions is just wonderful. And I think a really great path to understanding life at its fundamental level, and how cells fight for survival as individuals in the same way that together we fight as organisms.
Any other questions? Oh, actually, this is Safal here, who's actually a student at the 11 Lab, and he's one of the smartest kids I've ever met, so he's going to ask you something cool.
So one thought I've had, this is going to be somewhat peripherally related to your actual work, but it builds off the theme in the answer you just gave there. So one thing I've noticed about this more information theoretic view of biology is that the distinction between, bigger agents like human beings or large animals and cells acting as agents together to, do certain outcomes becomes somewhat blurred, right? Because less important is the actual scale of the organism we're looking at, and more important in a sense is a computational power or expressivity of the organism we're looking at. But of course, us humans have consciousness. If you have this collection of cells with, conflicting agencies and so on, and they give rise to emergent, sort of more complicated emergent behavior, I wonder how we can distinguish complicated emergent behavior in general and conscious complicated emergent behavior, and whether you have any thoughts on that. This is a massive question. So
Oh, wow. I'm surprised you haven't also brought in language models into that. We are in an incredible time, right? That we're finally seeing ways to approach consciousness, artificial consciousness. And what's surprising a lot of us, including by his own admission, Sam Altman, is that it's the exponential effect you get with linear scaling, and it sounds too good to be true that if you just build it big enough, it will come. But so far, it looks like that might be true. And I think what life basically is even from the molecule to the cellular level, and then the cell to the tissue to the animal to the society, is that effect, is that when you get enough of individuals, things together, emergent properties come from that. And now with AGI potentially. We see evidence of that. I think we'll see in the next 5 years that that's true. And this is the reason why Google is now building nuclear reactors. There's a race to be big, and big new things will happen. And the brain is the best example right now of that. Yeah, our brain's slightly different, right? There are parts of the brain that are different than a monkey, and there are some genes that create more contacts. But fundamentally, it's not that different. And there are other species with different types of brains, like an octopus, that have consciousness. And so we're not that special, but we do have a pretty compact, big brain. And this, size doesn't matter, it turns out, surprisingly. And so, I think that your question is a really good one, is what is it about consciousness and the interactions and the scale that seems to make all this possible? And what is it about that leads to emergent properties? I'm also happy to have you ask a second question given my answer, if there's more thought that you have.
I'll think on it a bit first. Let's see if anyone else has things to ask, and we can come back to it.
Elena, Vadim, maybe? Any of you in the back? Any questions? Nope. Last call? All right, that would be it.
No, who wants to?
Oh, he does? Oh, one more?
Yeah.
Go for it.
So what's your suspicion on a, say, quantitative or qualitative threshold after which these emergent properties actually emerge? Like, do you have any suspicion? Because we can say that it gets more complicated and after a certain point they come. But, having worked on this stuff for so many years, you must have some intuition about what kind of complexity we're looking at here. That's, I guess, what I'm interested in.
Do you mean intelligence or biology or aging?
Let's say, consciousness, intelligence, like that kind of threshold.
Gosh, well,
I'm asking for your personal view here.
yeah, well, the disclaimer is I'm just a yeast biologist. But I have been fascinated with consciousness. I think anyone who really stops to think should be fascinated by this. My simple view of consciousness, and I've explored my own brain with in India, and tried to find out what the heck is going on in there. And what I've come to, I wouldn't say conclusion. I should say what I think is going on is the more conscious you are is dependent on how easily you or how often you can observe the process of thought. And then you can observe the observation of the process of thought. And then you can observe that and observe that. And it, eventually, I don't know where that ends with computing, because that's also nearly infinite, the ability of a electronic mind to observe itself and even change its thoughts in the process. We're terrible at that as humans. Most of us never really gain control over our thoughts or our emotions. We try. There were some gurus and monks that managed somewhat, but most of the time we're just basically animals reacting to others and getting offended and getting upset. But yeah, true consciousness is the ability to watch yourself think and then watch that process. So I hopefully gave you something to think about. But I think computing is going to be fascinating to see, because it's going to not just reach consciousness in our lifetime, but have many higher levels of consciousness as well.
Thank you.
Awesome.
Very insightful.
Give it up for David Sinclair.
Thank you.
Hopefully we'll catch you next time in person.
Sounds good.
We'll see you. Bye, David.
Bye.